Okay, going to embryogenesis. When we want to create embryoids, okay, those tissues which look just like your zygotic embryos, okay, like remember, if this is a gynosium, no, then the pollen grain, the pollen tube will germinate, and then it will release one of the germ cells to fuse with the egg. Okay, then we get a zygote, no? Then we get a zygote. Then slowly it becomes into a seed, no? Forms into a seed, right? This is, let's say, the zygote. Then here this is the nucellus and all, no? So, somatic embryogenesis or embryogenesis is when we are going to create something similar to this embryo, okay, the one that you find in nature, similar to that, but they do not come from the male or the female gametes, they come from the other cells, okay, the diploid cells. I've already explained in the other chapter the organogenesis, so it's already easy, I think, for you. Okay, the next is embryogenic potential, means the capacity of somatic plant cells to produce embryos. Okay, the capacity of a plant cell means root, shoot, stem, etc. to produce an embryo type. Then an embryoid is a small, well-organized structure capable of or comparable to the sexual embryo, which is produced in tissue culture, of dividing embryogenic potential somatic cells. Okay, so an embryoid is a structure which is similar to the embryo that we find in the seeds. Okay, but this one is produced inside the lab. Okay, by using somatic cells, okay, by using any embryogenic potential somatic cell, means any cell which has the potential to become an embryo. Okay, let's look at the principles. There are two ways. It can occur directly without callus formation, okay. It can occur directly without any callus formation from pro-embryonic determined cells. Okay, means they're already programmed. No, they already know that I should become an embryo. For example, from the leaf cells of the orchid grass, directly we get the embryo without the callus. Callus is those mass of cells, no? But here directly we are going to get this heart-shaped embryo. So this one is the suspension. Suspension means it's inside that, remember that conical flask with the liquid media. This is inside the liquid, okay. Then figure B is what? It's starting to form these are all the embryos, okay. Directly we are getting embryo from these leaf cells. Mesophyll cell means leaf cells, and this is an enlarged embryo. Like this one is simply your heart-shaped embryo. This one only. So how did they do this? They took some leaves, okay, then they added this chemical dicamba, and then you culture the embryo, means you put it in the agar and you add these vitamins, minerals, hormones, and then you get the full plant. So as we said, there are two types of somatic embryogenesis. One, directly from any plant you get the embryoid, no, and then an indirect one. First, whatever explant you took, first it will go through the callus formation, then it will give you embryoids on the callus, and then you can produce the plants on transplanting and all. So direct means direct we are getting the embryo, Indirect means callus first, then on the callus embryo. Okay. So in the second type, we need to form some callus, and they and then the and then from those callus the embryoids will get induced. Okay, let's look. I think we better look in detail. For indirect somatic embryogenesis, where it has been induced under in vitro condition, means in the lab, okay, we have to use two different types of media. One to initiate the callus formation that will give rise to the embryoids, and the other medium to develop these into embryoids. Okay, so we need two media. Okay, first media is to produce callus. Second media is to induce from the callus to get embryos. Okay, so we need two types of medium. So in the first medium, it must have auxin. Okay, and in the second medium, we have to remove auxin or we lower it. But then in some cases, the first medium itself may give you the embryoids, but they cannot grow beyond. Okay, means they are not able to mature. Okay, so you have to do something. Okay, you have to add something so that that embryo will mature. Okay. And in the previous sentence, why did we lower down or remove the auxin? Is because they observed that there were some abnormalities, okay, in the embryoids when there was too much high level of auxin. So the embryo, they come out from the callus, okay, from the superficial callus, means the callus cells that are on the top, okay, not the ones from the inside. On the top, they will induce the embryoid. The embryogenic cells, they have a lot of cytoplasmic contents, okay, with lots of starch grain and a large nucleus and a dark nucleolus. Okay, so they are quite nutritive, means they are quite good cells that have given rise to the embryoids. So this image shows you the stage of embryogenesis. So I think you might have done this with in paper 7, no? 7 I think, or 8. Yeah, paper 7 I think you have done the embryogeny. So here these are the early stages of embryogeny. Then here it's starting to form the globular stage of the embryo. Then these are the later stages of the embryogeny. Okay, so this one you already know is the same diagram that you did for embryogeny. You did the globular embryo and the heart-shaped embryo development. No, it's the same. So during embryogenesis, when it's growing, they will go through 3 stages. First the globular stage, then the heart shape, then the torpedo stage. Okay, so here you can see, no, this one is the globular stage. Here, this one is a globular stage embryo. Then this is the sort of a heart shape. This is also heart shape. Then these are the later stages, which are the torpedo stages. Here, these are the globular. Here, this is just a more advanced image, real image, not a diagram. This is the real globular embryo stage. Then this one, the one you have seen when you did the dissection in your cucumber, this is the heart shape. And then these are the torpedo stage of the embryogenesis. Okay, so these are the real pictures.